case noisy people. Okay, so the rest of the genetic stuff that we need to go over for this course is in Chapter 9. Does anybody know what this picture is showing? That's the opener for Chapter 9. What process? Yep. Bacteria, yep, something's being transferred. This is a particular process. This is a particular structure that allows them to join up with each other that we've already looked at. Yeah. So there's a name to this structure that you should already know. Pili, sex papillus, right? So this process is called conjugation. That's one of the ones you need to know for this chapter. I didn't put in here the links like I've done in the past, right? Because at this point you guys should know how to get to them in your book for the animations. But I highly recommend the animations for this chapter because they're a really good summary of conjugation, transformation, transduction, and pretty good one on transposable elements as well. You know, if you've watched them before, they're short, they're well done, and worth your time. So, we may or may not have mentioned this previously. And I've used a term that you should know from biology class, but again, for some of us that's been a while. So does anybody know what haploid means? One set of chromosomes, right? And typically, how many chromosomes does a bacteria have? One. One circular chromosome, that's it. Unlike eukaryotes like ourselves, we're what? Diploid, which means we have how many copies of our multiple chromosomes? Two. One we got from our mom and one we got from our dad, right? So essentially, we have 2 copies of all the information we need. So if you get a bad copy from, say, mom, hopefully dad gave you a good copy. If both parents give you bad copies, this is when we end up with genetic disease, right, like sickle cell. Do bacteria have moms and dads? Not really, pretty much just mom. And again, just one copy. They have the copy of the cell that gave rise to them. So we know, right, that information in the DNA can be transcribed into messenger RNA and then translated into a protein. That protein gives that cell some ability, right? So in the case of sickle cell, anyone know what's wrong with the person's red blood cells? What are they not making properly? Because they don't have the correct information. What's the job of your red blood cells? Carry oxygen, right? And how do they carry that? There's something inside that cell that allows them to carry oxygen, right? Hemoglobin. Guess what? It's a protein. Where's that information encoded? How do you know how to make your hemoglobin? This is genetics, y'all. DNA, right? That information is in your DNA. So if your parents don't give you the right information, you can't make the right hemoglobin. Well, think about this. They only got one copy, right? So for sickle cell, you only get that disease as if both your parents give you a bad copy on how to make hemoglobin. If one parent gives you a good copy, you're good, right? No sickle cell. In the case of a bacteria, if they get a bad copy from their mama, they're screwed, right? No backup copy. No copy from dad. There's no dad. They're haploid. They have one copy. of all their genes for the most part. Of course, there are exceptions to that, but for the most part, 1 copy. Now, granted, they don't use the protein hemoglobin, but think of something else they may have, right? Not microtubules, but the proteins within their cells that make up their cytoskeleton, right? Or the ability to make the sex papillus, right? That's encoded in their DNA. Or to make a capsule. There's one you can think about, right, that we've talked about. How do they know how to make a capsule? That information is in their DNA. If they don't get the correct information on how to make it, can they make it? No. So phenotype is the physical expression of a gene. So if they don't have the information on how to make a capsule in their DNA, or they don't have the correct information, they're not going to make a capsule, right? Their phenotype is going to be different from others who can make that capsule. So whenever change happens for them at the DNA level, you usually see it at the phenotypic level, right? At that expression level. They can or can't do something based on the changes. And so we usually always see these changes because there's no backup copy, right? Where with humans, right, we've got 2 copies. So we really only see a phenotypic change if both copies are defective. Where the bacteria only have 1 copy, it's defective, then their phenotype's going to change because of that. So you can see that genetic change happens very rapidly in bacteria. And we saw that with that huge Petri dish, right? How quickly in 11 days you had mutated E. coli, I think it is, right, resistant to the antibiotic they were using. And if you read in this chapter, they talk about the experiment too that, mutation happens randomly, right? All the time. DNA polymerase misses its mistake, doesn't fix it. And so the organism changes. And then the environment kind of dictates, well, if that's good or bad, right? And whether they're going to predominate. They're going to select for. But mutation happens all the time outside of that pressure, right? It's when you have that pressure of the environment that you really see, right, the evolution, the change within the population over time. So bacteria specifically are really good models for genetic research because we only have to deal with 1 copy of all their genes, right? We only have to change 1 copy. But as we learned last time, they do have repair mechanisms, right? So if we change them so they can't do something, they could potentially mutate back to the ability to be able to do it. Especially if we only change 1 thing. So when scientists create and want to study mutations, they don't just create a single mutation. They usually create several mutations. At least 3. So that decreases the likelihood that they're going to spontaneously mutate back into their original form. When we work with auxotrophs, anybody do their reading? What does an auxotroph mean? So here's our mutants, and I realize this is kind of small. And there's a particular way they abbreviate things. Here, I can just make it bigger. I thought about making this one bigger, and I didn't do it. Should have known better. And I'll even crop it. I'll just look at one set. All right, so let's look at this. It says media containing both methionine and proline. Anyone know what those are? Right, good. They're amino acids, right? And we know we need amino acids to build proteins, right? So if you don't give them methionine or proline, they have to what? They need it, right? And you're not going to give it to them, so they're going to have to what? Make it. They're going to have to make it from something else, right? turn one amino acid into another? What do they need to do that? How do you turn one amino acid into another? You just wave your magic wand. Boop. How does a cell turn one molecule into another? No clue. Enzymes. 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 Are all together this morning for Friday. How do you turn one molecule into another inside of a cell? Enzymes, right? Which are proteins, right? Where is the information encoded in the cell on how to make an enzyme? All together now. DNA, right? In the DNA is the information to make an enzyme. Turn one molecule into another, one amino acid into another. Okay. What happens if you don't have that enzyme? That DNA is not right. You can't make that enzyme. You therefore cannot turn one amino acid into another, correct? So, this guy, he can, by the plus symbol, he can convert and make methionine, and he can make proline. So he doesn't care if you don't give it to him. He's like, I can do this. It's no problem. Give me something else. I'll turn it into these two. This mutant, on the other hand, is an auxotroph, meaning that it needs to have something provided for it because it can't make it. In this case, what can it not make? Methionine, right? Because you notice there's a little minus there. But it can make proline. So he's like, okay, I'm not so upset about the proline thing. But the problem is these are essential amino acids. So what will happen to that mutant with the fact that you didn't give it methionine? It's essential. You have to have it for life. Remember, it's the one that the start codon codes for. Basically, in bacteria, all their amino acids start with methionine. And he can't make it, and we didn't give it to him. What's going to happen to him? He's going to die. Yep, he's going to die. This guy, mutant number 2, right? He can make methionine. He's like, yeah, no problem there. But he can't make proline. Essential amino acid. He can't make it. We didn't give it to him. What's going to happen to him? He's going to die. The problem is, if we kill him off, how do we study him? Right? It's an interesting conundrum. So, as I said, they can spontaneously correct, go back to the wild type form. So say they can make proline and they can make methionine. So to get around this problem, we're going to usually create 2, usually 3 auxotrophs. We're going to change 3 things that they have to be able to do to live. So if we don't provide those nutrients for them, they will die. So we eliminate that problem if we just change one thing and they can fix it on their own. So for the most part, bacteria, single chromosome. Circular, right? One copy of all their genes. And a lot of them have plasmids. They don't have to, but a lot of them do. Another name for a plasmid is referred to as a replicon, because it can replicate on its own. And they'll keep more than one copy of a plasmid inside the cell. But they do monitor this. There's codes and information in that plasmid that help keep track of how many copies they have. and to make sure they get divided from one cell to the other when they replicate. Now, your book talks about how if they acquire a plasmid that's too similar to the one they already have, they're not going to keep it because they can't keep track, right? So I feel bad for parents of identical twins. Like, how do you keep track, right? You mark them somehow, don't you? I'm sure they do, right? They put like a certain color sock on a kid or something. I don't know how you keep straight identical twins when they're babies, right? They can't tell you, oh, I'm Angela. No, I'm, Sarah. Right? So right down to their code, plasmids can be differentiated from each other. And if they're too similar, it's too much of a problem for a bacteria. They're just like, okay, I'm going to get rid of this other one. It's confusing me. So the other thing that we'll talk about, especially more when we get to viruses, but we're also going to kind of mention it here as one of the gene transfers we're going to look at, is where a bacteriophage, these are viruses that infect bacteria, will inject their DNA into the cell, and it will incorporate into the cell's chromosome. So bacteria can have their own chromosome. In addition, they could have plasmids, and then even in addition to that, they could have viral DNA inserted into their chromosome.
So does the virus cause an infection in the DNA? I mean, in the bacteria?
I'm going to answer that when we get to viruses. Okay, so what are 3 ways plasmids differ from bacterial chromosomes? Because they are different, right? So they're both made out of DNA, right? Does their size differ? Which do you think is bigger, a plasmid or the bacterial cell's chromosome? You think plasmids are bigger? No, they're smaller. The chromosome is much larger. It contains all the information the cell has to have to live, right? Remember, plasmids are extra stuff. They're smaller. They're always smaller. But that's good in the sense that it doesn't take a lot of energy to copy them. They can easily be transferred from one cell to another, as we're going to see.
Is there a picture of that? Are there any photos of like plasmids?
Mm-hmm. In this lecture. I mean, basically, it's a big circle and a little circle. Right? So the other thing is, plasmids, they code for non-essential information. So does a bacteria have to have a plasmid? No. Right? Because remember, not all of them do, but a lot of them do. You have to have a chromosome, though, right? That's the essential information for a cell. But plasmids are non-essential. But are they good or bad? This is extra DNA, right? Good, why not? Anything extra is good, right? You go to the grocery store and they're like 2 for 1, right? Why not, right? And then they frequently have multiple copies per cell. We refer to this as copy number.
The plasmids do?
Mm-hmm. Where when a bacteria copies its chromosome, that's usually because they're getting ready to divide. They don't usually keep more than one copy of their chromosome. Of course, there's that exception we talked about last time where he keeps 4 to 10 copies, right, of his chromosome. That's rare, right? Most normal bacteria, 1 copy of their chromosome. When they copy that, they're dividing into 2 cells. Plasmids, on the other hand, they're going to keep a bunch of copies. And plasmids can be unrelated to that bacteria, right? That information doesn't have to be homologous or similar. Unlike chromosomes, like when bacteria want to share information that's on the chromosome, it's got to be similar enough that they can basically swap one piece out for another. And you'll see that a lot as you watch the animations for this chapter, or as we look at the different stuff. We're like, okay, I got this DNA, but now where am I going to put it? Right? I got to find a place to put it. So, of course, when we're studying mutants, we want to be able to isolate and detect them to be able to study them. The most easiest isolation methods are direct methods, where you can directly select for just the mutant. So ones that are resistant to an antibiotic, that's really easy to select for, because the ones that aren't resistant, what could we do to them? So there's a bacteria that's resistant to, say, streptomycin. How can we get just that one that's resistant to streptomycin and not deal with any of the others that are sensitive, killed by streptomycin? What you think? How do we get the one that's resistant? We don't even need to go that far. What does streptomycin do to bacteria? Most bacteria. It kills it. So if we put streptomycin in the media, put the bacteria in, who's going to survive? The one that's resistant, the mutant that we want to study, and we're going to kill off all the ones that don't have antibiotic resistance to streptomycin. Wow, that was easy, huh? That's great if it's something like that, but what happens if you're dealing with an auxotroph, where if you take away histidine, say, you kill it? Well, crap, I wanted to study him, but I just killed him. In that case, you have to do something like replica plating, or another technique they talk about in your book called patch plating. So, as we said, something like streptomycin, right, where it helps us select for the mutant, we could very easily just grow them in the presence of streptomycin and get just our mutants that are resistant to streptomycin. That's a nice, easy way of directly selecting for a mutant. That's all well and fine if it's the mutation is something like that where it can allow us to select. Not always the case. Here's another one where we could directly select, right? You can see, oh no, this is the one, this is the problem. Okay. So on this plate, right, you see what? 9 different colonies, 3 by 3. These 2 are mutants, right? And you can see them. They are colored differently. So we could directly select for them. So this is another example where we could just take those colonies off. Clearly they've changed, right? The wild type, everybody's white, but these guys are red. But if we take these same colonies and we put them on other plates, this is under a particular set of conditions. This is a different set of conditions. You notice when you compare the 2 plates, we don't have 9 colonies on this plate anymore, right? The colony that should be here and here are gone. Whatever that condition was that we were growing them under, it killed them. They don't have an ability that the rest of the wild types, the normal guys, have. So how are we going to study them if we killed them off? You do what's called replica plating or patch plating. You start with growth media and everybody grows. Mutants and non-mutants. You can't tell the difference. You use almost like a stamp. It's a piece of felt, and you know felt is kind of fuzzy, that's sterile. Right? So there's no microorganisms on it, no viruses, nothing. You press it onto this plate, and then you can use it to stamp the same orientation of colonies on several other plates. And each one of these plates is a different growth condition. So then you can compare, right? But you're always going to stamp an original one where everybody grows. And usually what they don't have here, they usually have a line on these plates, right? So you match it up. So you make sure you know the orientation. This is where patch plating is a little bit better, where they use a grid on the bottom of the plate, and so they put the colonies in a specific grid on the plate so they can keep track of who's where. on each plate. So now when they look at this plate, everybody's growing, but this plate, you can see there are 2 colonies that were on the original plate that are now missing. So you know that these, right, are your auxotrophs. Whatever condition on that plate stops them from growing. So you know you're going to have to provide that condition to grow them to be able to continue to study them, right? You got to go to this plate, though, and pick them up and go put them in that condition now, right? Say put them in media containing histidine, something they can't synthesize on their own anymore. Does this make sense to you guys? Right. So as we're trying to screen for them, we kill them. So we have to have an original plate where we can actually pick them up and grow them and study them and give them that nutrient that they can't make on their own anymore. So the Ames test, which isn't covered in your book, unfortunately, is a test that's designed to be able to study whether a particular chemical is a mutagen, if it'll change DNA, and therefore be a potential carcinogen, cancer-causing agent. So they work with a particular strain of Salmonella that has a mutation. It can't make histidine. So histidine has to be provided for it or it will die. Now, of course, this is a single mutation, right? And it can spontaneously revert back to not needing histidine. But we know the rate at which that happens. It's a relatively low rate. So if you look at this plate, right, just a few of them spontaneously mutated back and can grow without histidine on this media that's lacking histidine. If they put the chemical substance on, and you get all of these colonies on here, that means that that chemical you put on there mutated them back to not needing histidine at a faster rate than just spontaneous mutation would happen. And then, of course, you have your control, right, where some other liquid that doesn't contain the mutagen. This is done a lot, right? So if we have a new chemical a chemist comes up with and say they want to put it as a food additive, they're going to run through the Ames test. If it mutates the bacteria, is it going to end up in our food? God, no, right? If it's mutating bacteria, God only knows what happens to us. So the people that get really upset about animal testing, they love this test, right? Because the cute and fuzzies don't get tested. But it passes the Ames test, it still gets tested on the cute and fuzzies, right? Because the cute and fuzzies are more closely related to us than bacteria. But the good news is less cute and fuzzies have to get tested if we do the Ames test first. You know what I mean when I say cute and fuzzies, right? Animal testing. Okay. Okay. No, if they pass the Ames test, then they're still going to get tested on animals to make sure they're not going to hurt humans before they get released to be put into, say, our food. But if it fails the Ames test, it's not going any further, right? It's a mutagen. We're not going to obviously test it on cute and fuzzy. We already know it's bad news. Where before, everything had to be tested on cute and fuzzies, right? Just to be sure. Make sense? Okay. No one complains about us torturing bacteria, though. I don't know the date on the Ames test. I can look it up, though. I would say probably in the 70s. It's been used a long time. So what we have left on mutations was in this chapter. So we have talked about all the rest, right? But we hadn't talked about transposable elements or transposons and knockout mutations. Those are discussed in this chapter, chapter 9. And so transposons were discovered by a scientist, actually a female scientist. Anyone know who? Barbara McClintock. And she noticed this natural phenomenon that happens in corn. Specifically maize corn, and I got a picture because people never knew what I was talking about in the past. On the next slide. So what these are, is what transposition is segments of DNA that are mobile. They move from one place to another. So they can move within the chromosome, they can move from the chromosome to a plasmid, from a plasmid to a chromosome. So sometimes these guys are nicknamed jumping genes, right? Because they literally move, right? Jump from one place to another. And so there she is, and she was studying corn that has, notice that each one of these is a different color. Each one of your corn kernels is an embryo, right? That would give rise to a whole nother corn plant. So genetically they're very similar, right? They came from the same mom, and whatever little spermy pollen granule went down the silk and got to it and fertilized. But why is it that we're seeing all these different colors? Well, when she investigated it, come to find out, there were these segments of DNA that were moving from one part of the chromosome to another. The problem is when they moved from one section to another, they didn't just like insert themselves in between two genes. Sometimes they literally jumped into another gene and disrupted that gene. So that's what I'm trying to show you in this picture, is that, you know, these are, say, this is the chromosome, say, because it's bigger, right? And here's a plasmid, say. And here are 3 genes, 1, 2, and 3. That are part of a transposable element or a transposon. They jump, they come off of here and insert into B, right? So here they are in B. So they just moved from the chromosome into the plasmid. The problem is they just split B into 2 pieces. Whatever B coded for, is that cell going to make any more? No. RNA polymerase doesn't know that B has been split into 2 pieces. It's not going to jump from one section to another. No, it doesn't work that way. So now you just disrupted that gene. Basically made it non-functional. So depending on what that gene did, in the case of the corn, it kept disrupting or activating or genes that were related to color. We use this in laboratory research, molecular research all the time now. This ability to be able to insert into genes and basically knock them out, right? We can specifically say, I don't want this gene to work anymore. I'm going to stick this other thing in here so that cell can't do it anymore. And I'm going to study on how does that affect that cell. They've done this not only at the cellular level with bacteria, but to the organismal level. So they've taken mice and literally knocked out an ability to be able to study it at the organism level. So there was, when I worked at LSU and did research, in the immunology lab, there was a lady, and I remember them talking one day, the caretakers were talking, and they were all concerned about these knockout mice. And I was like, what's the big deal about these mice, right? Why are they so obsessed about how clean they are and making sure none of them die and, you know, they're well kept and nobody goes in there and messes with them? They're mice. I mean, I understand we were doing experiments, you know, that experiments are important, right? But why were these mice, like, got the, you know, rock star treatment? Well, it was because they had a genetic knockout mutation. And that's not an easy thing to achieve, especially back then. This was like 15, 16 years ago. So this was a very expensive line of mice to produce, right? And very important to her research. absolutely. Most people don't like this particular, like the maize corns, like these, they're not as sweet, like these really colored ones. This is sometimes also referred to as Indian corn, like this was the corn that Native Americans grew, right? We've modified extensively corn since then, genetically. Don't get me started. But yeah, you can eat it. It would be non-GMO corn, probably. Non-genetically modified. Okay. So I was really excited that in this book they had this diagrammed out, because I used to have a crappy little drawing that I tried to do to explain to my students what I used to do in the lab. So we utilize the ability, and there are restriction enzymes out there that were discovered that microorganisms have, and it was probably for cutting up foreign DNA, like when viruses inject their DNA, the bacteria could cut it up and it wouldn't affect them. It was like a defense mechanism. Well, the really cool thing about these restriction enzymes is they actually read code and they cut specific codes in DNA. And the way they cut it, they don't cut both strands equally. You have these overhangs. And so then you can like piece together pieces of DNA using these restriction enzymes. And so scientists do this all the time now in molecular labs. And so there's a naming system, you know, where these restriction enzymes were found originally. So for instance, this one, the Echo R1, was discovered in E. coli, this particular restriction enzyme. So they've made these, genetically engineered these plasmids, these circular pieces of DNA. And they have, within a gene, these restriction enzyme sites. So these codes of DNA that will be cut specifically by these restriction enzymes. So scientists can go in and they can take this plasmid and they can cut it with the enzyme, and they can cut pieces of DNA they want to study with that same enzyme and put this stuff together in a test tube, and their DNA that they're wanting to study will sometimes incorporate right into this plasmid. Pretty cool. But the thing is that this ability, this enzyme, when you stick something in here, you're doing an insertion, right? You're basically knocking out that gene. It can't make that enzyme anymore. Well, can we tell if a bacteria can't produce an enzyme anymore? Sometimes, depends on what that enzyme does. In this case, it takes a substance known as X-gal, and I want to get it wrong. Oh, yeah. It takes X-gal and it cleaves it and makes a blue compound. So here is the enzyme. Right? It'll cleave it and it'll turn your E. coli blue. So any of the guys that have an intact plasmid are blue. All the E. coli are blue. If the insertion works, they can't make that enzyme, right? They can no longer break X-gal, which is a clear substance, and break it in such a way that now it turns blue. So your transformed or changed or cloned E. coli are now white on this plate. The other thing that's on this plasmid is an antibiotic resistant gene. E. coli is normally affected, killed by ampicillin, which is a penicillin derivative. If they take in this plasmid, because that's the other important step, so we make these plasmids, right? Then we stick them in the bacteria. If the bacteria take it up, then now they can make the enzyme to destroy ampicillin. So guess what I have in that plate so that I only select for the bacteria that took up the plasmid? Ampicillin, right? So I'm going to select for just the ones that have the plasmid. And then amongst them, I only want the white ones, right? because that means that that X-gal enzyme, that ability to break X-gal and turn blue, has been disrupted. Because they're not turning blue, are they? They're staying white. Kind of crazy, huh?
You know how you said that sometimes the genes could jump and it will it will kind of mess up the DNA.
Mm-hmm.
Can they jump? Like, can the DNA recognize the mistake and put it back? Like, this is I don't want you to be.
No.
So it's just going better.
It could move again. The jumping gene could jump back out. That could happen. But there's no enzymes that go along and recognize that, oh, something got inserted into here and I need to fix it. No, it doesn't work that way.
So it's like you don't really realize it's gone.
Right, I guess, yeah. Well, except for the fact that if it's something you need, right?
Yeah.
If that gene that just got knocked out or disrupted, if you need it, then yeah, you're going to know it.
Okay.
Like when you get pickpocketed, you don't know when it happened, right? You know after because you have no money.
Yeah.
Make sense? Okay. Trying to make real world analogies. Good.
That's how I learned.
That's what you got to do. Tell yourself stories, right? That you can relate to. So, we've already talked about transformation a little bit because whose experiment that he wasn't actually trying to discover what he discovered, which was transformation, but he also proved that DNA was what caused cells to have these abilities, right? Remember what experiment that was? It was with the mice and Streptococcus pneumoniae. That's that man, yeah. The Griffith experiment. And so this is a little bit different diagram than the one I gave you previously. This isn't one in your book. But I like it. It's a pretty good summary. So remember, some of them could produce that capsule, right? And it killed the mice. They died of pneumonia. And then the unencapsulated ones didn't cause infection, right? The capsule was pathogenic, that feature. And he found if he killed the encapsulated ones, of course. If he killed the bacteria, it didn't kill the mouse. And then he decided, well, these two didn't kill the mice, so if I put these two together, right, should be even better vaccine. No, instead his poor mouse died. And then when he autopsied, he got back out living encapsulated cells. Well, how did these live cells that don't know how to make a capsule figure out how to make a capsule? They didn't figure it out. They stole the information, didn't they? Because these dead cells contain DNA, do they not? They do. And putting those dead cells in the same environment of the living cells picked up that DNA and they used it. They found a spot in their chromosome that was homologous, close enough, right? Probably what happened is these guys are a mutant strain from the original strain that it's encapsulated, right? Something happened to their gene that allowed them to make a capsule. Some mutation happened. So they had a similar section, and they just swapped it out for the one that could make the capsule. So I highly suggest, because I'm going to go over these relatively quickly next time, watching the animations for these three processes: transformation, transduction, conjugation, transposable elements. And then we're going to start viruses next time, so they'll be very shortly. I'm not going to put a time frame on it, but definitely before I lecture on it, another lecture on viruses, which I didn't look up what chapter, but it's in the syllabus. Imagine that. What's the next chapter we're going to? So you want to know, I'm going to look it up right now. 5 and 8? Yeah, 5 and 8. Thank you. Chapters 5 and 8 are up next.